Great. Thank you, Erica. Good evening, everyone. My name is Mary Ann Collins, and I work in Martin Lab over in Building 68. And there we go. In the Martin Lab, we're interested in the process of morphogenesis, or more simply put, how developing tissues form their shape. Now, you can think of this process as analogous to the art of paper folding, known as origami, which transforms a flat sheet of paper into different shapes through a series of precise folds. And much like origami, living tissues deform and undergo these massive transformations that will shape the developing embryo from a simple collection of cells into a complex three-dimensional organism. And depending on how that tissue is folded, can give rise to many diverse body plans of various shapes and sizes. Now, much of what we know about the mechanisms that drive tissue morphogenesis and these massive tissue deformations has been done in studies using the Fruit Fly Drosophila melanogaster. And by combining those classic Drosophila genetic toolkit along with advanced imaging techniques, we can use the Drosophila embryo to watch life fold before our eyes. Just over the course of a few hours, this embryo will undergo dynamic, there we go, thank you. This embryo will undergo dynamic transformation through a series of folds, which will transform the embryo from a pseudo-flat tissue sheet into multiple layers. In the Martin Lab, though, we are interested in understanding the formation of the first fold to form, which is known as the ventral fold. And this involves the inward movement of a subset of cells along the bottom edge of the embryo shown here. So tissue movements like this and others that occur are driven by mechanical forces that are generated by each individual cell within the tissue that then must work together to collectively bend and ultimately change the morphology of the embryo. So to understand the impact that these mechanical forces have on cellular behavior, I take images of embryos at different stages of development and then track how cells change their shape and organization within the tissue during the folding process. So shown here, each cell is labeled here by its nucleus shown in gray, and they are physically linked to one another by these cellular junctions shown here in orange. Now, using this junctional signal, I can then trace and segment out each individual cell within the image in order to track how they change as the tissue evolves. So if we look at this first pair of images here, prior to tissue folding, cells within the entire tissue sheet have similar morphology. Yet as the tissue begins to fold, we now see these cells take on new shape, with those in the center now constricting and shrinking in size, while those at the outer edge begin to stretch. And as this tissue continues to fold inward, those stretched cells that were once on opposite ends of the tissue now come together and form new contact, resulting in a newly formed ventral fold down the middle of the embryo. Now, by using this comparative view, we can really see tissue origami in action. The left side of the image shows this newly formed ventral furrow that forms, illustrating that the new tissue is adapting and transforming into a new morphology. The segmentation on the right allows me to track these different cellular behaviors and observe how cellular mechanics can contribute to the overall reshaping of that tissue. Together, they paint a dynamic picture of an organism truly undergoing a massive remodeling event to help the developing embryo take on its new form. And with that, I'd first and foremost like to thank the organizers of the Koch Image Awards, namely Erica, and the panel of judges for selecting my image and giving me this incredible opportunity to share with you all a glimpse of my work. I would also like to thank my advisor, Adam Martin, the larger Fly community for developing and sharing all these wonderful resources with us, as well as the NIH for funding our research. Thank you.